that produce a cellular response. The hormone receptor is a protein that has 7 hydrophobic transmembrane spanning regions that project through the cell's plasma membrane. The receptor is associated with a G protein that is comprised of alpha, beta, and gamma subunits. The peptide hormone assumes its active configuration, fits onto the receptor binding site, and conveys its message. The message results in a conformational change in the receptor that is transferred mechanically to the G protein. Mechanical activation of the G protein results in the alpha subunit replacing guanosine diphosphate with guanosine triphosphate. The activated alpha subunit separates from the beta and gamma subunits and migrates within the plasma membrane to an inactive phospholipase C enzyme. Energy captured from the hydrolysis of the terminal phosphate of guanosine triphosphate is transferred through the alpha subunit to activate the phospholipase C. The process of activating the phospholipase C converts the guanosine triphosphate back to guanosine diphosphate, and the alpha subunit rejoins the beta and gamma subunits to reform the inactive G protein. Activated phospholipase C degrades phospholipids that make up the plasma membrane to produce 2 second messengers, inositol 1,4,5 trisphosphate and diacylglycerol. The inositol 1,4,5 trisphosphate is released from the membrane and binds to a receptor on the endoplasmic reticulum. This intracellular signal results in the release of calcium stored within the endoplasmic reticulum. The released calcium plus the diacylglycerol from the membrane activate an inactive protein kinase enzyme in the plasma membrane.
And that then goes on to the protein kinase C. So what I haven't learned is to get out of this thing. Sorry. All right, so as we look at going back now to, I should just copy that slide in here, but going back to this pathway, so we've looked now at 3 different mechanisms by which G proteins interact. Okay? And all 3 of those have second messengers. So now let's look at hormone receptors that are directly linked to the enzyme without the G protein. Okay? So G proteins were first, and now we're going to look at 2 examples of that where they're not directly linked.
Thank you.
That's a phospholipid in the membrane. And it's going to be split to form the IP3, and also called triphosphate and the diethylglycerol.
Does it matter the spelling of triphosphate and triphosphate?
Maybe for chemists, but so what do you notice different about this particular illustration? We still have a membrane-bound receptor. All right, so there is well, it's not just that the GTP isn't already connected up there. There's no G protein for it to be connected to. Okay? So that's why this was not listed under a G protein pathway. So this is probably going to happen a little bit faster, because we're not going through the earlier steps. But we're also missing areas of control and regulation. So we have to regulate the presence of the receptor and the enzyme without having G protein in there to be able to regulate. So if a cell uses this pathway, any pharmaceuticals that affect G protein won't be effective. Right? This is going to be directly activating the enzyme. So again, here we have a hormone produced by the heart. Most of us don't think of the endocrine system, endocrine organs, including the heart. But H1 natriuretic peptide is a hormone we'll pick up again when we talk about the heart and about the kidneys. What chemical do you think natriuretic is referring to? Sodium. Chemical abbreviation for sodium is Na. All right, atrio refers to the atrium of the heart, which is the receiving chambers of the heart. And this is for free. I get, again, you will need to know it when we get to the heart. But if blood volume is too high, that's going to make blood pressure too high. And it's going to stretch out the atrial walls. And so if they are overstretched, that stimulates the muscle cells to release atrial natriuretic peptide, which as a hormone goes through the blood to the kidneys and causes the kidneys to become diuretic, to release more urine, more water in the urine, and that lowers blood volume and lowers blood pressure. So you can see how the heart would, it's helpful for it to be involved in blood pressure regulation in that fashion. All right, so it binds to its receptor, which is not a channel, all right? And then the enzyme guanylate or guanylate cyclase will interact with the receptor. And notice some similarities. This is not my picture, but it should have cyclic GMP right here. So instead of having adenylate cyclase converting ATP to cyclic AMP, we have guanylate cyclase taking GTP and converting it to cyclic GMP. Very similar as far as that is concerned. We just have a different amino acid involved, guanine instead of adenine. And so downstream, that's the role of the enzyme, not the role of cyclic GMP. Downstream, cyclic GMP then results in the phosphorylation. Enzymes become activated. So essentially does, or we can go backwards, phosphorylation or dephosphorylation. So essentially it's very similar, same intracellular changes that cyclic AMP would occur. And in fact, the same enzyme that breaks down and degrades cyclic AMP breaks down and degrades cyclic GMP. Phosphodiesterase. Just a much quicker event than that involving G proteins.